Thank you so much to Dr. Jackson. We're very privileged to have him coming and speaking to us. He's the neuroscience scientist and founding director of the Community Access, Recruitment, and Engagement Research Center at Massachusetts General Hospital and Harvard Medical School. So we're very privileged to have Dr. Jackson with us today, and he's going to be doing some talking about brain changes and healthy aging with Alzheimer's disease.
Yes, ma'am.
So thank you so much for coming.
All right. Okay. Thank you, Valerie. All right. So before I get started, can everyone hear me okay? If I speak about this speed and about this volume, everyone's good? Yes. All right. Great. So we're going to go ahead and get started today. So I'm going to talk to you about the brain, aging, and memory loss. And we're going to try to get through a few key points today. But before I begin, I always want to talk about this next slide. So this is called the disclosures of my conflicts of interest. This is a complete list of everybody that gives money to me or gives money to my research center. So if anyone is ever talking to you about something that you don't know about, then you need to understand where their revenue streams are coming from. So if I happen to say something nice about Mass General, or if I happen to say something nice about the Michael J. Fox Foundation, you can say, well, maybe, it's just because they're signing some of his paychecks. So it's always good to make sure that you know where those dollars are coming from. So I'm not here to say anything nice about any of these organizations, so I don't think you have to worry about that today, but it's just something to keep in mind for the future. If you're ever learning about something that is new to you. So we're going to try to get through 4 key points today. First, I want to talk to you about how memories work and where they're stored in the brain. Then we're going to talk about what goes wrong with Alzheimer's disease and some of the very, very latest research. I think the latest research today is from October 28th, so still pretty recent here. And then finally, we'll spend our last few minutes with information on how to age gracefully. Things that you can start doing right now today to lower your risk of Alzheimer's disease, okay? So those are the 4 key points we're going to try to get through. Does that sound good? All right, excellent. Let's get started. So the first thing that we're going to try to do is talk about how memory works and where memories are stored in the brain. So when we think about memory, most people start off by thinking about something like this. There's like a projector or a video camera or Netflix inside of your head. And the thinking is that anytime you want to call up a memory, you just sort of hit rewind to that particular day and time. You press play, and then you can sort of see it with your mind's eye. Well, there's one thing that we've learned. There's a lot that we still don't know. There's one thing we've learned in about 125 years of memory research, and that this is wrong. Memory does not work even remotely like this. So when we have those senior moments, when we walk into a room and we can't remember why we're there, if we can't remember where we put our keys or that really important piece of paper, it's because, and we get frustrated, it's because we often think that memory works like this, and it doesn't. So if that's not a good metaphor for memory, why don't we think about a better metaphor for memory, all right? So this is the metaphor that I'd like you to keep in mind. Does everybody know what Legos are? Does everybody recognize what those are? Okay, great. So I have a 6-year-old daughter, so I'm used to stepping on Legos at home rather than with being able to interact or play with them. But Lego is a much better metaphor for how memory works because you have to build a memory in order to store it inside of your head, and then you have to rebuild that memory from a bunch of different pieces all over the brain in order to retrieve it. So that gives rise to 2 really interesting phenomena. One is that as we practice rebuilding those memories, as we start doing that process over and over again, it makes those memories stronger over time. So older memories, those well-practiced memories, you're really used to building those Lego pieces, that Lego memory, over and over and over again. So it's a relatively strong memory. It's pretty robust, is what we call it. The second thing, which seems counterintuitive, but is also true at the same time, is that these memories very slowly change over time. So every memory, every time you retrieve it, it's affected by the way that you're remembering it in the current moment. So if you have a very happy memory, but then you're a little bit sad when you're remembering it, it sort of colors that memory just a little bit. if you're very angry, or if you're somewhere new or different, all of those things play a role in helping change the trajectory of how that memory forms over time. So the long and the short of it is, if you had a really awful argument with your spouse back in 1994, you really do both remember it quite differently. And that's just the way that memory works over time. So both of you have a very strong, very robust memory of the same event that is fundamentally different. So memories go stronger over time, but they also can change over time as well. So now that we know a little bit about how memory works, and a little bit about why memory is better off as a Lego, let's talk about where memories are stored in the brain, okay? So what I've got here is a big, colorful map of the brain. So first of all, the brain does not actually look like this. It's not blue and yellow and green. All right, some of you are laughing. That's a good sign, all right? So you're well on your way to becoming duly deputized neuroscientists. So I'm what's known as a cognitive neuroscientist, which means that I only care about these colorful parts of the brain. These parts that are in black and white, this is the cerebellum and the brainstem. They regulate things that I don't think of as very important. So, you know, like breathing or your heart rate or any involuntary functions of your vital organs. I don't care about any of that because I'm a cognitive neuroscientist. So I only care about these big colorful parts up here. So I'm going to give you a quick whirlwind tour of the human brain. So what we've got here is the frontal lobe. And just to orient you, this is a side view of the human brain. It's a sagittal view. So it's basically somebody looking this direction. Their eyes would be right here underneath the blue part. So the frontal lobe is right here behind your forehead, and it's called the frontal lobe because it's at the front of your head. So neuroscientists are very, very intelligent people, but we are not a creative species. So the frontal lobe regulates thinking and personality and attention. So hopefully each and every one of you are really engaging your frontal lobes right now to listen to what I'm saying and not necessarily mind-wandering to where you parked your car or what your left foot in your left sock feels like at this moment, which you weren't thinking about until I mentioned it, and now you are. Next to the frontal lobe, or just behind it, up here at the top and the back of your head, is called the parietal lobe. And the parietal lobe does a lot of different kinds of things. It's a real workhorse of the brain. It plays a lot of different functions, but one of the key roles of the parietal lobe is that it integrates the different senses together. So hearing and touch and smell and taste puts them all together into one cohesive experience. And so for the philosophers in the room, if there is a root of human consciousness or conscious experience, scientists believe that it would be found here in the parietal lobe. So in the back of the head, it looks small, but it's actually a quarter of your brain. In red, we have something that's called the occipital lobe. And the occipital lobe is dedicated to one thing and one thing only, which is seeing the world around you. It might feel a little ironic that what you used to see is at the back of your head, but that's just the way that our brains are organized. And then finally, we've got here in green a part of the brain that's called the temporal lobe. It's next to your ears, so you might think that it's important in hearing, and it is. It's home to something called auditory cortex. It's also home to 3 brain regions that are going to be very central in the remainder of our talk, okay? So for those of you who are taking notes, you might want to write this down. So the 3 regions are the hippocampus. So who's heard of the hippocampus before? Show of hands. Okay, great. So that's off to a good start. So the hippocampus. Now, I wonder how many of you have heard of this next region. It's called the parahippocampal formation. No one's ever laughed before when I've said that. But so you've got the hippocampus, the parahippocampal formation, and then the third region that I want to teach you today is called the entorhinal cortex. All right? So I'll repeat those 3 regions. The hippocampus, the parahippocampal formation, and the entorhinal cortex. So for those of you who are taking notes, those 3 regions form a super region that we call the medial temporal lobe. So medial just means medium, like in the middle of the temporal lobe. So the middle of the temporal lobe is filled with these 3 regions: the hippocampus, the parahippocampal formation, and the entorhinal cortex, okay? So try to keep those in your brain because we're going to come back to them a little bit later on. Now, the medial temporal lobe, those 3 regions that I talked about, are where memories are reassembled. It's where all the Lego pieces converge to create that Lego memory that you're trying to remember later on. okay? It's also the part of the brain that is the most susceptible to age-related change, okay? So you might think that, well, as I get older, you know, my body doesn't work quite as well as it used to, so I figure maybe my brain doesn't work quite as well as it used to. It turns out that your brain changes with age, but it doesn't change all in the same way. So going back to the frontal lobe has a kind of a steady decline over time. So it's biggest, and this is kind of the bad news, it's biggest when you're in your early 20s, and then it's just downhill from there at a relatively steady decline, all the way through your 80s or 90s. So you might think, okay, that's probably just what the brain does, but it's not. So if we also compare that to the age-related change we see on the occipital lobe, that's a relatively steady size your entire life. It changes maybe a teeny tiny bit, but less than a fraction of 1% between when you're 20 and when you're 90. So that part of the brain doesn't really change with age at all. So we've talked about the frontal lobe, which has that steady decline. We've talked about the occipital lobe, which doesn't seem to have any decline at all. So what happens with that medial temporal lobe? What happens to that hippocampus? It has what we call a curvilinear function with age. Curvilinear is just a fancy word that means curvy line, curvilinear. Again, neuroscientists, not creative. So the curvilinear function means that this part of the brain stays about the same size through your 20s and your 30s and your 40s. And then when you move into your 50s, it starts shrinking. And in your 60s, it shrinks a little bit faster. In your 70s, it shrinks a little bit faster. In your 80s, it shrinks a little bit faster. And then when you get to your 90s, you're probably fine, because something else is going to get you first. So there's this increasing deceleration, what's called an accelerating decline over time. So this is where senior moments come from. This is why when you move into your 50s and 60s, you have those memory issues that you didn't before, okay? So the long and the short of it is you can take a deep breath if you have some of those senior moments, and you can say, I'm fine, because it's not Alzheimer's disease, okay? It's just a normal part of how memory functions and how memory works. There's no part of your brain that is more sensitive to the process of age than the medial temporal lobe, the hippocampus, the parahippocampal formation, and the entorhinal cortex, okay? So we've talked about memory. We've talked about where memory is in the brain. We've talked about how that changes with normal aging. So now let's get to the reason why everyone's here. What happens in Alzheimer's disease? And that's what we're going to talk about next. Before we talk about the details, the nitty-gritty of Alzheimer's disease, I wanted to go through the leading causes of death in the United States. So this is for the year ending 2016. Normally, the Centers for Disease Control update this data every year, but under the current administration, they don't have as much money as they used to, so we're stuck with data that it's 2 years old instead of 1 year old. But what you can see on this list is that Alzheimer's disease is the 6th leading cause of death in the United States. There are a lot of other causes of death that are kind of much more prominent. Heart disease and cancer are number 1 and 2 by a long way. But a few actual points about this. Alzheimer's disease is 6th now, and it has been for some time. But as recently as 1999, it was 10th on this list. So it's really, really moving up. And I can tell you that in calculating the rate of death and how that jumps year over year, so I update this as soon as the new data are released every year, or I guess now every 2 years. And what you see is that the number of deaths that are attributable to any one cause may fluctuate by between 1% and 3% every year. So heart disease goes up and down by about 1 to 1.5%, same does with cancer, accidents, stroke. Between 2014 and 2015, Alzheimer's disease increased by 11% in 1 year alone. Okay, so that's not just because of better diagnostic rates. It's partly to do with a lot of kind of the graying of Americans' population. So a lot of the baby boomers are moving into retirement age, and when you move into retirement age, you also move into what we call the zone of increased risk for Alzheimer's disease. So you're much more likely to develop Alzheimer's disease too. And then in the year ending 2017, the CDC, for maybe the second time in recent memory, actually revised downward the life expectancy for the average US adult. And part of that reason was due to the opioid epidemic that is really ravaging America. But there was a second reason why they're revising it downward is because of the creeping specter of Alzheimer's disease. So, we actually dropped about 1/10 of a year between 2016 and 2017, in part because of Alzheimer's disease. So we really are facing a looming public health crisis when it comes to AD. And so now we're going to talk a little bit about what that means. One question that I often get is, I don't quite understand the difference between Alzheimer's disease and dementia. Is it possible for someone to have Alzheimer's disease but not dementia? Can they have dementia but not Alzheimer's disease? This chart is one way of trying to help clear that up. So dementia is this umbrella term. It just means that you have problems with memory and thinking in a way that interferes with your everyday life. You're just having some kind of difficulty getting through the day compared to the way that you used to be. Now, the leading cause of dementia, especially for older adults, is Alzheimer's disease. And we'll talk about what that means formally in just a moment. But there are lots of other reasons you can have dementia, as you can see on this chart. So you can have dementia due to Parkinson's disease. You can have dementia due to vascular factors. You can have something called Lewy body dementia. You can have dementia due to severe alcoholism or depression. In some cases, the dementia is reversible with treatment. In many cases, it is not. The leading cause of dementia, the leading cause of thinking and memory problems is Alzheimer's disease. Okay? So, yes?
Could you just briefly talk about vascular, since that's also quite a large area?
Yes, so vascular dementia and then additionally mixed dementia tends to mostly be a mix between vascular and Alzheimer's disease. So you can actually count this as part of the blue as well. So vascular dementia really refers to problems with the blood vessels in your brain. So this can be triggered by a stroke or more commonly by what are called mini strokes or TIAs, transient ischemic activities or attacks. Thank you. So these mini strokes can occur in different parts of your brain. So we talked about it earlier. So they tend to be much more prevalent in the frontal and temporal lobes of the brain. And what happens is it affects your memory and thinking in tiny ways. So you'll have one of these little, kind of transient events, and you will see kind of a step down in terms of how someone's able to function. And then they'll be relatively stable. Then they'll have another event, and there'll be a step down. So it's really like this stair-step function that happens slowly over time. So depending on your demographic and socioeconomic status, the vascular dementia time course can be upwards of 2 decades, but usually it's associated with a slow decline in your cognitive ability over time. Now, where this occurs depends on where these mini strokes are occurring in your brain, but they tend to happen disproportionately in the frontal lobe and the temporal lobes of the brain. So you will see less, you can still see memory problems, you'll see less of those. You're more likely to see problems with concentration, some changes in personality. You're likely to see perhaps some difficulty in thinking or reasoning. So for example, if you happen to be an accountant for 35 years, but then you suddenly couldn't balance a checkbook, those kinds of events that you might see might be attributable to vascular dementia. Yes, sir?
Are you going to talk about physical brain injury as well as related to these, like football players?
So I'm happy to talk about CTE at the end of the talk, but this talk is really going to be focused on Alzheimer's disease. Hopefully I'll be able to come back, and usually when I come back and do second talks, I delve into the different triggers and causes of dementia. So I'll give a much longer discourse on vascular dementia. I definitely talk about CTE because Boston is actually home of the world's best and most advanced CTE research center and Boston University. I'll talk to you about Lewy body dementia, Parkinson's, and frontotemporal dementias as well. So I can talk a little bit about that during the Q&A, but this talk is really focused on Alzheimer's disease. So when I say that the talk is going to be focused on Alzheimer's disease, this is what I'm talking about. So before we talk about what's on this image, let's talk about what's not in this image. So this is not a picture of somebody getting lost. This is not a picture of somebody forgetting, their spouse's name. This is not a picture of somebody putting their keys in the microwave. This is what we call a blown up silver stain of the human brain. So it's a very zoomed in slice of human brain tissue, all right? And what you see on this slide is that there are 4 circles, 2 red, 2 black. And these refer to the 2 primary proteins or peptides that are deeply involved in the Alzheimer's disease process. This is how Alzheimer's is defined from a research framework. This is how it's defined from a diagnostic framework as well, okay? So when we talk about Alzheimer's disease, we're talking about this physical change that happens in the brain. But what is this physical change is probably what some of you are wondering. I don't necessarily know what peptides or proteins are or how they go wrong or what's affected in Alzheimer's disease. So let's start with the ones in red. So the ones in red are called amyloid. Who's heard of amyloid before? Amyloid, A-beta, AB42. It goes by a variety of names, and they refer to different parts of the pipeline. But generally, we're talking about a problem with a peptide, which is a mini protein, called amyloid. So the way we think about it is that amyloid is something that you have in your brain your whole life. It's really deeply involved with the transport of lipids, little fats, around your brain. It zips around like Uber or a taxi. However, it goes a little bit wrong in a way that I like to kind of give this example that I made up during one of these talks actually on the fly. But does everybody know what Saran wrap is or cling film? Okay, good. So normally, if you're watching a lovely commercial about Saran wrap or cling film, what is that process like for the glowing homemaker who's pulling it out? It's a very straight, very shiny piece of plastic material. It tears off perfectly. It forms a tight, perfect seal over the potato salad or the leftovers from the barbecue. They sort of put it in the fridge and they basically like float away because they don't walk on the ground because they are not like a mere mortal or anything like that. What's the reality of Saran wrap? What is that process like? It's a pain in the neck. So why is it a pain in the neck? Because it doesn't do anything that they say in the commercial, right? It sticks really well to itself when you're trying to tear it, and then it doesn't stick to the bowl when you're trying to put it off. And then when you're trying to get it to tear off the teeth, it won't do it in the right way, and it gets tangled up. Yeah, and if you're cheap like me, then that's like 3 cents you're not getting back, right? You basically have to try to find a way of disentangling it from the yellow box, and you have to throw it away. Well, a very similar process happens in the brain when it comes to amyloid, except you can't throw it away. It just keeps building up in between the brain cells. And it forms what are called amyloid plaques. And so amyloid plaques they're bad stuff, they're pretty dangerous, but left alone by themselves, amyloid plaques don't cause a whole lot of damage. They're a hallmark symptom of Alzheimer's disease, but they need more than that to get going. So in addition to amyloid, you see the black circles up there refer to tau. Now, who's heard of tau in the context of Alzheimer's disease? Tau tangles, neurofibrillary tangles, something along those lines. Now, tau is the second protein that seems to go wrong in the brain. So tau is really important. Again, it's something you have in your brain your whole life. Its main job is maintaining the rigidity of the cell walls. So it keeps those cells strong, so the stuff that's inside stays inside, the stuff that's outside stays outside, right? During Alzheimer's disease, tau becomes what we call hyperphosphorylated, which means it has a phosphate group attached to it for the chemists in the room, the organic chemists. What that means in reality is that the tau becomes weakened. It becomes poisoned in a way. And a metaphor for how this works is, have any of you ever had water damage to your home, or seen what water damage looks like? So if you have water damage on one of these walls, you've got like a big bulbous stain coming out of it, it's turned brown. Can you just sort of paint over that? Can you put a picture over it and it's fine? Usually, if you are lucky, you have to replace that drywall. You have to replace that sheetrock, right? Because the wall has been weakened by this water. The same thing happens when a phosphate group gets added to the tau. It weakens the cell walls. So the brain cell almost unravels. So when this brain cell is unraveling from the inside because of the tau, and you've got this poisonous amyloid Saran wrap coming in from the outside. They combine together and they trigger what's something that's called apoptosis in the cell. So the cell sort of has a mini seizure and dies. So you might think, well, okay, Jonathan, this is a very complicated process to kill one brain cell. You know, as a human, I know that I have 70 billion brain cells, so one or two, it's not that big of a deal. However, let me show you what this process looks like over time.
What triggers the phosphate to attach itself to the tau?
So what triggers the phosphate to attach itself to?
Yes, and where does the phosphate come from?
So the phosphate actually comes from relatively unknown sources. However, it's thought that they may be a byproduct. This is kind of a fringe theory, but it's gaining more traction. It's thought that it's a byproduct from some energy dysregulation that happens at the metabolic level within the cell. So basically, for those of you who know a little bit more about biology, and I promise I can't spend too much time on this, unfortunately, is that the mitochondria, if you remember from high school, is the energy powerhouse of the cell, right? And that's probably the only thing you remember about the mitochondria. It's also a very, very susceptible part of each and every cell. And so when it becomes dysregulated, usually because of the presence of free radicals, you might remember free radicals from the 90s in terms of like, your health. But it turns out they can kind of jump in and to a part of the brain cell or any cell in the body, dysregulate the energy structure, and then one of the byproducts that happens from the energy dysregulation process is the increased presence of some of these phosphate groups within the brain in particular. Now, these phosphate groups tend to have a high affinity for the tau protein in general, and so what happens is initially the tau gets, I don't have this picture anymore, so the phosphorylated tau tends to be concentrated along the axons and along the dendrites of each cell. But as Alzheimer's disease becomes more severe, it moves up into the cell body, where it then starts to interact with the amyloid peptide. So sorry, that was a little bit in the weeds for everyone else in the room, but basically these phosphate groups work with the tau, move up into the cell, and make the cell much more susceptible to the effects that we see with amyloid. So when you have this process building up over time, you can see that it has a huge effect on the brain. As you move from left to right, you can see that there's a healthy brain on the left, and then hopefully all of you can appreciate how the brain is losing its brain cells. It's shrinking. You can see it by these looking at these sagittal or these coronal views of the brain. And then all the way on the right-hand side, you can see this is a coronal view, so this is looking at somebody's brain straight on this way. You can see what a healthy brain looks like compared to a brain that's been deeply affected by Alzheimer's disease. And the regions of the brain that we've been talking about the most, the hippocampus, the parahippocampal formation, and the entorhinal cortex here are the parts of the brain that are the most affected by this process. So one of the reasons why Alzheimer's disease seems to affect memory is that it attacks those parts of the brain first: the hippocampus, the parahippocampal formation, and the entorhinal cortex. So just as those parts of the brain are very sensitive to normal age-related changes, they're also deeply sensitive to the changes that we see that are associated with Alzheimer's disease. So this is why sometimes you'll go to your primary care doctor and you're saying, I have memory problems, and she'll shrug her shoulders and say, well, that might be Alzheimer's disease or it might be normal, because we know that both processes, both aging and Alzheimer's, tend to target the same parts of the brain. Okay, so now we're going to talk very quickly about some of the latest research in Alzheimer's disease. I think all of the information that I'm here to tell you about is less than 6 weeks old, or it's been updated within the last 6 weeks. So all of this is brand new stuff. So before I talk about that, a question that I often get about the latest and greatest in Alzheimer's disease is about genetic factors. And before we get into the weeds of that, and I'm happy to talk a little bit about the genetics, and especially the epigenomics of Alzheimer's disease, we need to understand what genetics looks like in kind of an everyday context. Genetics are probability-based by and large, which means that there's a chance that something will happen to you, but it's tough to know what that chance is, and it's tough to really translate that chance into practical action. So all of that probably just sounds like words. So I'm going to give you another example of how we use probabilities in an everyday setting. And I think today is the perfect day to talk about that, which is the weather. So if I were to renounce my ways as a neuroscientist and declare myself Channel 5 weatherman Jonathan Jackson here on the scene to tell you that there is a 10% chance of rain, who's going to take their umbrella with them? Who's going to take a coat, a raincoat, maybe a slicker, a parka? No? Nobody? Oh, okay. All right, Valerie. All right, we've got one taker here. All right, 10%. All right, I like it. Okay, so basically most people say no, but one person does say yes. Now, if I were to come back and say, you know what, hang on, I'm very new at this weatherman business. When I said 10% chance of rain, I meant that there's an 85% chance of rain. So who's going to take their umbrella with them? Who's going to take a parka? Okay. All right, so most people are going to take it, but not everyone. Some of you are really going to play your odds, okay? I think there was an 85% chance of rain this morning, and so now here we are. So again, most people will take up those odds at 85%, but not everyone. So here's where we live. Here's where we live when it comes to genetic risk of Alzheimer's disease. When we talk about, if I were to say, Channel 5 weatherman Jonathan Jackson, there is a 35% chance of rain later this afternoon. Who's going to take an umbrella? Who's going to take a raincoat? No one. Oh, okay, so a couple of people. All right, good. We've got one person who might take part of the umbrella. I think that's what this means. Maybe you'll take just exactly half of the umbrella. But when we're in that zone of 35%, it means that it's like, well, I mean, I would like to know more information. I'm going to look outside. I'm going to, lick my finger and put it to the wind to see what's happening. Anything to move that 35% up or down, because we're deeply uncomfortable with that 35%. And when it comes to risk, lifetime risk of Alzheimer's disease, many of us are in that 35% zone, where some of us will be affected and many of us will not. And it's just hard to know right now, with the information that we have, where people are going to be. So if you have a question about family history of Alzheimer's disease or the genetics of Alzheimer's disease, the best answer that we can give right now is maybe. You're probably in that 35% zone. Okay? So I know that is a deeply unsatisfying answer, but that's where the most robust science is right now. We are working really hard on polygenic risk scores in the Alzheimer's space, and maybe in about 2 or 3 years' time, we'll be able to talk about that with a little bit more clarity. But right now, I'm sorry. We're going to be stuck with talking about focused ultrasound scanning, which is something I've been talking about at these talks for many years. I remember the first time that I brought up this slide on focused ultrasound scanning. It was actually in 2014. It was a talk that Sarah had arranged. And I said, does everybody know what focused ultrasound is? So for those of you who are familiar with ultrasound, you kind of encounter it with pregnancy, or if you're maybe looking for a particularly difficult to detect cancer in your kind of thoracic cavity. It turns out that you can weaponize this technology in a way that helps you fight the battle against Alzheimer's disease. You can trigger the ultrasound waves to the resonant frequency of either amyloid or tau. So we learned a little bit earlier that this tau or the amyloid in particular, is this protein that builds up inside your brain, and you can't break it apart, you can't throw it away, you can't get rid of it. Using ultrasound technology, you can break apart that amyloid protein, that amyloid plaque, into pieces small enough that they can be cleared out through the body. So what's nice is, again, when I first presented this a little over 4 years ago, I think I was over like in Topsfield or something, and I said, this is a really exciting technology. It worked on each and every one of the 10 rats that they had currently done this study on. And then the audience kind of groaned because I was like, well, sorry, they haven't even found that 11th rat yet. However, things have moved very, very quickly in this space, and that's because they haven't had to go through the normal FDA approval channels to make this work. And what has happened is this is the picture from May 2nd, 2017, when they enrolled the first human subject in a clinical trial looking at focused ultrasound therapy as a therapy in the fight against Alzheimer's disease. That trial closed in December of 17. July of 2018, they reported that the ultrasound therapy was tolerated very, very well. And they've launched a phase 2 study at the end of September 2018 in 4 sites. The prime site is in West Virginia. They haven't announced the other 3 sites yet, but I will let you know as soon as I know. But what they found is that in the 4 years they've tested it in rats, pigs, monkeys, and now people. So what's really exciting is that it seems that ultrasound therapy is an effective tool in the fight against Alzheimer's disease. What's really, really great about it is that it requires no pharmaceutical intervention, no drugs. You're just opening up this kind of small hole in the blood-brain barrier to clear out the amyloid plaques, and then it closes right up again. There are no side effects. And what we've seen in a lot of the animal models is that not only does this get rid of the amyloid, but the rats and the animals have actually been able to recover memories that were previously thought to be lost due to the mouse models of Alzheimer's disease. So we're not yet at a point where we can see those changes in people, but in the next year or so, we will start to find out. Yes?
How can you make rats and pigs and monkeys get bad enough to be experienced?
So you can actually genetically engineer animals to have Alzheimer's disease, or the animal equivalent of Alzheimer's disease. So you can make sure that they have the plaques and tangles in their brain in the same locations, in the same ways that we see them in people. And so we've actually gotten really, really good at giving and curing Alzheimer's disease in animals a lot. The tricky part is in trying to kind of replicate that same success in people once we've seen it in animals. But we've been able to do that for some decades now. Yes. Yes, ma'am.
What is the diagnostic technique now? It used to be post-mortem.
Yes. Strictly speaking, right now, officially, it's still a post-mortem diagnosis. However, the diagnostic tools that we have at our disposal now, especially in conjunction with the research context, mean that we can almost completely approximate the accuracy that we see at postmortem. So we're like around 99.5% accurate in vivo, so before people die, at knowing whether the dementia that someone is experiencing is due to Alzheimer's disease or some other factor. And so that's because we've gotten better at neuroimaging, so we can do things like MRI or PET scanning to look for evidence or changes that are associated with the presence of amyloid and tau in the brain. So we're pretty good at doing it in life, but again, the gold standard is and will always be postmortem. So focused ultrasound scanning has really come a long way in the last 4 years. They're now opening, like I said, a second study in the United States, which started, again, just about 6 weeks ago. So not too long. This is a really interesting article that was published, I think about 3 weeks ago. Yeah, about 3 weeks ago, about subtypes of Alzheimer's disease. Now, this is something that has been rumored for a long time, that Alzheimer's disease doesn't quite work the same way in everybody. But it was thought for a long time that most people who developed Alzheimer's disease looked one way, and a very, very small percentage of people had a completely different presentation. However, what they did in this particular study by Young and colleagues did a machine learning study. They used artificial intelligence to classify the progression of Alzheimer's disease in about 1,300 brains. They spent a couple of years on this study. And what they found was that there were 3 distinct subtypes of Alzheimer's disease. And what was interesting about these subtypes is that they were roughly equally seen across these 1,300 brains. So a third of them had this typical staging that where I said it starts off in the medial temporal lobe and then moves into the frontal lobe and then back across the parietal lobe. But there are 2 other subtypes where the progression looks very, very different. What you see here is that in this particular stage, you see that it starts off in the frontal lobe and the temporal lobe at the same time and then moves back. And then subcortical means that it starts kind of deep inside the brain and then slowly moves out over time. So it's interesting that a third of the brains were seen in the typical stage, a third were seen in cortical, a third were seen in subcortical, which means that for the first time we're starting to understand that Alzheimer's disease may not be one thing. It might be at least 3 different things that we have previously thought kind of got all confused together. So no wonder why we're having such a hard time, trying to cure this thing, because we've been confusing these different subtypes with each other. So this needs to be replicated in a much larger sample. It would be great to see this in about 5,000 or 6,000 brains, which we have. But for now, it looks like the way that we think about Alzheimer's disease might be changing in a really, really big way moving forward. And then what I wanted to talk to you about, spend most of my time talking to you about, is about a drug called BAN2401. Has anyone heard of BAN2401? Okay, so those of you who are writing notes, and those of you who are a little bit Google savvy, you might want to follow up with this and keep an eye on this particular story, because it's probably one of the highlights of 2018. So something that I often say in these talks is, boy, we're getting really, really close to a cure or a treatment for Alzheimer's disease. And I've been saying that for many, many years without sort of a clear indication of what a cure would look like. BAN2401 is our first glimpse at what an effective treatment in Alzheimer's disease looks like. So let me tell you a little bit about the story about BAN2401. Yes?
Spell it with the B-A-N,
And it's 2401, the numbers.
and then
20.
401, right, with it.
It's all sort of one alphanumeric word, correct. BAN2401. This is jointly developed by Biogen and Eisai. And so last December of 2017, they came out a little under a year ago and said, BAN2401 doesn't work. It's one of the many, many, many drugs that just doesn't work in people. We really tried. We're so sorry, you guys. We looked at 850 people over 4 years, and it just didn't work out. Aw shucks. And then they came back in July and they said, hang on guys, no, we were totally kidding, we were totally wrong. It so worked. It worked so well that we didn't even understand how well it worked. And it turns out they had run 2 different sets of analysis. For those of you who are kind of curious about this, the first round of analysis was Bayesian. The second round of analysis was kind of traditional frequentist. And we're using the traditional frequentist analysis. They announced July 3rd, like right before a national holiday. They said, we promise it totally works. We'll tell you the more details later. And then they didn't give us any more information. It was literally a 1-paragraph press release saying, totally worked. We promise. We'll give you more details later. So then they waited until the last day of the International Conference on Alzheimer's Disease, AAIC, which was in Chicago this year. The last morning of the last day, they packed everybody into a room the size of an airplane hangar and said, all right, here are some details. We found that there was a 93% reduction in amyloid burden and for individuals who were taking the highest dose of this drug. And we saw a 47% degree of slowing of cognitive decline. Now, when we talk about a cure for Alzheimer's disease, it turns out that we actually have to define what that means. And the Food and Drug Administration in the United States has said that you have to meet 2 endpoints to be deemed a treatment for Alzheimer's disease. 1, you got to take care of amyloid. You have to show that you are reducing the amount of amyloid in the brain. And you can do that without actually opening up somebody's head by doing a PET scan. And then number 2 is you have to slow the rate of cognitive decline. So they could still be declining in terms of their thinking and memory abilities, but it has to be a slower rate of decline compared to somebody who is not taking the drug or is taking a placebo. So if you look at these data, they reduced amyloid, they got rid of almost all amyloid, and they cut the rate of cognitive decline in half. So BAN2401 is thought to have met the primary endpoints for a treatment for Alzheimer's disease. So we're there. Now, this was an extremely complicated design. If you Google it, you'll find out that they did some really unorthodox, weird things that might have skewed the results. So for a number of months, people were saying, wait, we're not totally sure that you cured it. we want to look at, a couple of other things. We want to understand, why some groups where some people were not represented across all the dosage rates and things of that nature. But they gave an update at another clinical trials conference at the very end of October, so like right before Halloween, where they went and they kind of combed through the data and they said, all those weird things that we found, we went through and we talked about them, it doesn't make the effect go away. It's still there. So people who are taking the highest dose of this drug saw, if anything, a slower rate of cognitive decline. So they argued that this 47% rate was conservative, that it might actually be 60 or 70% reduced rate of cognitive decline. So again, because it's still very, very complex data, we're still trawling through it as a field. But if true, and it looks like upon a second and third round of analysis, this may be true, we have all the makings of a real-life therapy that would be effective against Alzheimer's disease. So really, really good news here. Yes?
So is this still in clinical trial, or is it starting to become available?
So we still need to work through the data. But this was a phase 2 clinical trial. It would still need to go through a little bit of phase 3 testing and then submit for FDA approval. So depending on timelines and how aggressive the drug makers are, well, maybe not 4 years, maybe like 2 years before this could be approved by the FDA.
With tau.
Great question. No. So the round of drugs that are currently being announced in clinical trials are mainly focused on reducing amyloid burden. It's only recently that we've introduced either sort of tau-focused therapies or combination therapies. Those won't report out from clinical trials, at least at the phase 2 or phase 3 level, until the early 2020s.
I'm trying to recall my memory.
Yes, sir.
It's not quite there yet. But I read something years ago that it's not just the amyloids that will cause, and that even if you had a reduced amyloid content, you still could get Alzheimer's. Now, is that no longer true?
So it's partly true, but it's one of those things where it's a little bit more complicated. It's certainly true that in order to experience the death of brain cells or a lot of the cognitive decline, you need amyloid and tau in the same space at the same time. So the thinking right now, the thinking in these early clinical trials is that, you know what, the tau is there and it's bad, but it's not specific to Alzheimer's disease. It's not specific to any disease. So anyone over the age of 20 has at least some tau burden, irrespective of whether you go on to develop Alzheimer's disease. And then anyone over the age of, I think, 80 has a significant tau burden in their brains. So the thinking is, let's clear out the protein or the peptide that is unique to Alzheimer's disease, which is amyloid. And so that's sort of like the way that scientists talk about this is a bit gauche, but I'm going to use the same metaphor. It's like taking the bullets out of the gun. So the tau is still there and it's still dangerous, but it's no longer able to cause any damage. So the thinking is if we can remove the tau burden, the amyloid burden, we don't have to worry about the tau. Now, that question, that assumption is not quite right. It's one of those things that's like a B+ effort. So now drugs that are entering clinical trials testing are focused on only removing the tau or removing amyloid and tau together to see if that's more effective. So we're moving into this age where it's not just a matter of can we cure Alzheimer's disease, but what's the best therapy for myself, for my presentation of the disease and my family. So my guess is that all three of these kinds of approaches will work, removing the amyloid, removing the tau, or removing both. It's just a matter of what would be the most effective for any particular clinical presentation. Yes, sir?
Is there anything comparable being done for the other forms of dementia, in particular frontotemporal, and are they also caused by amyloid?
No, so amyloid is unique to Alzheimer's disease. Tauopathies are pretty pervasive across the entire dementia spectrum. So the thinking is that if we get good at removing tau in Alzheimer's disease, we might be able to extend those findings across a range of the other kinds of dementias, especially frontotemporal dementias and especially Lewy body dementia, which has a distinct isoform of tau. In Alzheimer's disease, there are 6 isoforms of tau that are present. In Lewy body dementia, there's only 1 or 2. So the thinking is that if we get good at removing tau in Alzheimer's disease, we'll be able to extend those to other disease areas as well. Yes, Sarah?
Can you just address again, like the Ariceps and all that, how the
Sure.
difference between that and what?
Yeah, so there hasn't been any new drug approved for Alzheimer's disease since 2004. And that class of drugs were anticholinergic therapies like Aricept and Namenda. These do not affect the disease course at all. They don't affect amyloid, they don't affect tau, they don't affect the death of brain cells. Taking Aricept and Namenda is like taking a cold medicine or a flu medicine. So those of you who have had to take these before, you know that that cold or flu medicine doesn't cure your cold or flu. It just makes you feel better. It helps you function out in the world for a time. And so over time, things like Aricept and Namenda or combination drugs, they don't work as well anymore. So on average, it buys you about a year and maybe not a good year. So Aricept and Namenda, the drugs that are currently approved, aren't terribly effective. And even when they are, it's just about alleviating the symptoms rather than the underlying disease. Namenda, N-A-M-E-N-D-A, because it affects the NMDA receptors. It's an anticholinergic therapy. So these days, doctors are more likely to prescribe Aricept than Namenda, or more recently, they'll prescribe some kind of extended release Namenda. Yes, sir.
What are the rules, the FDA rules, whatever, about before the work that has to be done, and maybe more than one trial, maybe independent studies or something, before it can actually be released for public use?
So there are 4 distinct phases on the FDA trajectory. Before you even talk about the FDA, you have to go through a drug discovery process. You have to test this at the molecular level. It has to make a lot of sense. You have to do animal testing and animal model work. That takes, on average, 4 to 7 years. And then you can begin what are called phase 1, or sometimes, nowadays, they don't call them phase 1 anymore. They're now calling them first-in-human studies. And so that is a small safety study where you'll test it on about 10 people. So when I talked about the focused ultrasound testing, that was a phase 1 study. They tested it on 7 people in Canada, in Toronto. And that's usually what's called a safety study. Can they take the drug without being harmed? They're not looking for any kind of clinical endpoints. They're just making sure that it's tolerable. Phase 2 is usually what's called a dosing study. So they're saying, can we give them a lot? Do we give them a medium amount? Do they give them a little bit? What kinds of side effects do you see at the different dosages? And again, is it relatively safe? And if all of that works, let's start to take a peek at those clinical endpoints to see if it affects the things that we want it to affect. So that will have, depending on the phase 2 study, it might have upwards of 20 to 50 people in it. Then there's a phase 3, which is kind of a long-term efficacy study that's usually done over a 2 to 3 year period. You'll enroll usually several hundred or a few thousand people to those clinical trials. And that way, you're looking specifically at whether the drug affects the clinical endpoints in the ways that you care about. So you know a little bit about side effects, you know about basic safety elements. Does this drug affect the disease or disorder in exactly the way that we want? That has to be predefined by the industry sponsor and the FDA, and once it does that, they present all their paperwork for approval, and then it goes into phase 4, which is post-approval. It's available for purchase, but they still continue to monitor for any kind of unforeseen effects.
So this has still quite a long road to go.
So they've wrapped up their phase 2 testing. They might go through an abbreviated phase 3 period, depending on how robust these data are. But this can actually be they have started to introduce some accelerated processes within the FDA based on what's happened in cancer. So they may not have to go through a full FDA phase 3 clinical trial to meet the approval criteria, because they did a relatively large phase 2 study because there were 850 people, which is almost at phase 3 level. So the odds that this will move through, given the context of other drugs that have been almost as effective as BAN2401, is pretty good. So it's not like we're putting all of our eggs in this particular basket. There are 4 other therapies that are similarly promising in the Alzheimer's space. They are all focused on amyloid right now. But we are going to be moving into phase 2 and phase 3 testing of combination therapies probably in 2019 or 2020. So we're really moving into this golden era of possible, potential, very promising therapies for Alzheimer's disease. Okay, so it's 2:01, and I had a few more tips and tricks that I wanted to advise you on, but I'm out of time, unfortunately. So I wanted to say that, there's currently no cure for Alzheimer's disease, but ongoing clinical trials, especially in phase 2 and phase 3 testing, are getting awfully close, awfully close. So I think that the time is very, very short before we can announce a promising therapy for the disease.
What is band 2401? What is the structure?
So most of the structures for the amyloid therapies are monoclonal antibodies that tend to attack the amyloid structure at one of several stages. So one is like when it's first forming those fibrils, and then all the way up until it attacks the full-on amyloid plaques. So the particular structure of band 20401, which is largely proprietary, but they've released a couple of drawings of what it looks like, and it looks like that its mechanism of action is basically adhering itself to the very early fibril structures. And then basically, in the way that of most monoclonal antibodies, it sort of raises its hand and flags the immune system to come and clear it out, even though it is relatively insoluble. So band 241 only affects the early aspects of the process. We have not had good luck looking at trying to attack the amyloid plaques once they're fully formed. There's some reasons as to why. One thinking is that it might be too late in the process. Two is that the amyloid plaques, this is going to be a bit of a head trip, the larger amyloid plaques may be in some way minorly protective in the brain, as opposed to the dangerous fibrils which are floating all around. So it's really like either like the protofibrils or the fibrils themselves that are mostly targeted by band 2401, and currently it's thought that is the primary mechanism of action for the drug. That it is, the proof is in the pudding that we do need to go early in attacking those fibrils with the drugs. Yes?
So would it be ever, we haven't talked about money and drugs at all, but prophylactic use of that kind of, if it only attacks early form?
So I think band 2401 will not be used prophylactically. So this set of clinical trials has been in individuals with mild to moderate Alzheimer's disease. There's a chance that you could use it for somebody who's at the mild cognitive impairment stage, but there's actually a separate class of monoclonal antibodies that are custom designed for prevention purposes rather than intervention purposes. And so some of those do tend to be more aggressive in attacking the more fully formed amyloid plaques, because those are the most dangerous, and those are a little bit less plentiful in individuals who are pre-symptomatic as opposed to those with a formal diagnosis. So like I said, there are many, many different kinds of therapies that are just coming over the horizon that may be promising. Some of them will be used in kind of prevention therapies. But right now, where we're seeing a lot of really interesting action is in the early disease stages, before there's too much damage to the brain. Yes?
Is Alzheimer, is a fatal disease, or people die?
Yeah, so that's
something else?
Right, that's a great question. Is Alzheimer's disease fatal, or do people die of something else? Most times, it is not fatal. Most times, something else will get you. And if you read about famous people in the news that died of Alzheimer's disease, they always use the same expression, which is that somebody died of complications due to Alzheimer's disease. This is the case with many neurological diseases, so not just Alzheimer's. But usually what happens is the disease process kind of goes through the brain, and it really starts to affect function. It really starts to affect motor function and your ability to move around. So later in the disease stage, people stop moving around as much. And what often happens is they get a respiratory infection and they can't cough. They get pneumonia and they can't clear it from their lungs. And they'll die of pneumonia or a respiratory infection. So that's usually what gets people with Alzheimer's disease. Although it is possible that the disease process can go all the way to the level of the brainstem and stop your brain's ability to perform involuntary functions like breathing or so swallowing or even your heartbeat, although that is very, very rare. Yes, ma'am.
My sister-in-law kept my father-in-law alive with Alzheimer's for over 2 years
Mm-hmm.
on a breathing tube and a feeding tube. I never asked the details, but who actually pays for that? To keep someone alive that is mentally gone. They can move him around in the bed, the bed's
That's
still-
true, yeah.
Basically, he was a body, just
Mhm.
rotated, but he lasted over 2 years before he died.
Yeah.
Who picks up the bill? Medicare, I have Medicare. Who picks up the bill for that?
So, I mean, it depends on, right, so the question is who pays for some of that very intensive acute care late in the stages of Alzheimer's disease? So she mentioned, that a loved one was cared for 2 years with a breathing tube and a feeding tube to ensure that these weren't going to be the triggers or the cause of death. The truth of the matter is that Medicare can pick up some of those expenses. Insurance, especially long-term care insurance, can pick up some of those expenses. But depending on who you are and your financial situation, some of that could come out of pocket. And it gets pretty pricey, especially if you have staffed care 24, 18 hours a day. So it really depends. Sarah, did you have a thought?
Can you just touch for them too after this, just on the Mediterranean diet?
Sure. Yeah. So unfortunately, I didn't get to all the happy news, all the positive aspects of this disease. But I wanted to talk about one thing that people can do, which is maintaining a healthy diet and weight. The Mediterranean diet I assume everybody's at least heard of it. The Mediterranean diet has been robustly shown to lower your risk of Alzheimer's disease. So there was one very large study done in 2015 that showed that individuals who stuck to the Mediterranean diet for about 4.5 years cut their risk by about 57% of developing Alzheimer's disease. And those who were only moderately good at the Mediterranean diet, so they occasionally snuck off to McDonald's, they still cut their risk by about 1/3. So what's nice is that this has been replicated in a lot of different ways, and it's relatively well validated at this stage. So I think it's enough to actually make a clinical recommendation that if you can do the Mediterranean diet, please do the Mediterranean diet. And I think what often comes up is, what is the Mediterranean diet? I think you know it's on the Mediterranean diet. So it's all of the things that you know that you should be eating, all of the things that have been recommended for decades, cutting out the red meat, cutting out the animal fats, cutting down on the sugar, all of those things that we know are bad for us, cutting down on the carbs and the processed foods, trying to eat more fruit and nuts and fresh vegetables, all of those things on the Mediterranean diet. Fish, a little bit of poultry is good things. Nuts are good, but please don't only eat nuts. You're not squirrels. But a couple of things that you might not have known, you can have red wine, a glass a day. You can have dark chocolate in moderation. So there are some perks to the Mediterranean diet, but again, it does seem to be largely positive. Yes, ma'am.
Sorry, I have to go meet with somebody, but is there more that you weren't able to give some of the stuff? I mean, I have other questions, like, for example, what's early Alzheimer's versus later Alzheimer's, early onset, et cetera. Is there another place that I can go and get more like what you might be talking about?
Yes. So the website for the Alzheimer's Association, alz.org, has a ton of information. Most of it is very, very accessible. It's easy to read and understand. And they talk about a lot of things. They actually have a couple of web pages on the difference between early onset, late onset, younger onset, later onset, and kind of differentiate all of those things. And they kind of differentiate between stages of onset and the different stages of the disease process and all of those things. But again, if you want to know more, then talk to your local programming coordinator at your friendly Council on Aging. I'm happy to come back. Usually when I come, I can give you some information about Alzheimer's disease, but there's so much going on in this space, there's so much going on in the dementia space, that usually it's best for me to come back 2 additional times, so 3 talks, where you set the tone, you let Valerie, you let someone know what you want to hear about, and I kind of craft a talk around those things. So it sounds like if you want me back, I'll talk about the different kinds of dementia and how those present and what those are like and what the latest research is on those. And if you want to know more about Alzheimer's disease, I'm also happy to come and do that as well. So I'll have to stop there, unfortunately, but thank you so much.
I think we will definitely ask Dr. Jackson to come back.
Yeah, I'd be happy to.